of DNA.
Okay, so deoxyribonucleic acid, one of the fundamental aspects of human genetics. It codes all of our information and genes that makes us who we are. Isn't that right, Austin?
That is correct.
Thank you. So I'm here with Austin, and today we are going to instruct you guys on the structure of DNA and the specifics on how DNA replicates itself. The structure of DNA revolves around the nitrogenous bases, which are adenine, thymine, guanine, and cytosine. These four bases fall under two categories: purines, which contain two rings and are larger than the other category, pyrimidines, which only contain one ring, as you can see in this picture. The purines are adenine, as you can see, which has two rings, and guanine. Pyrimidines are the other two, thymine and cytosine. The reason adenine only pairs with thymine and guanine only pairs with cytosine is because of their hydrogen bonds right here in the middle. Adenine can only have 2 bonds with thymine, while guanine can do 3 bonds with cytosine, and they cannot mix each other up. This pairing is also known as Chargaff's rule.
DNA has so many carbon molecules that scientists decided to order them into categories called primes. DNA has 2 important primes that are 5' and 3'. 3' is seen as the start of the DNA and can have more nucleotides added to it, while 5' is seen as the end of the DNA strand and cannot have more nucleotides added to it. The DNA strand is also seen as anti-parallel because they are parallel to each other or run in opposite directions. The 5' carbon on one strand is opposite to the 5' carbon on the other strand and causes the DNA strand to end opposite of each other. According to the diagram, the phosphate that is shown comes from ATP, which gives energy to the DNA strand.
So now that you know the basic structure of DNA, let's talk about how it replicates.
According to the diagram, there are 3 origins of replication that form bubbles in the DNA. DNA helicase is an enzyme that unwinds the DNA and forms a replication fork, separating the 2 strands and making them available as template strands. Topoisomerase is a protein that breaks, swivels, and rejoins the DNA. It helps relieve strain to the double helix ahead of the replication. Single-stranded binding proteins are proteins that bind to the unpaired DNA strands during DNA replication and stabilizes them and holds them apart while they serve as templates for the synthesis of complementary strands of DNA.
Once the parent DNA is unzipped and the origin of replication has been placed, a leading strand and a lagging strand is formed. The leading strand is the easiest to replicate, as a primer is placed and DNA polymerase III follows the chain and replicates the DNA. It brings an incoming nucleotide triphosphate to the 3' of the new strand and releases 2 phosphate groups to create enough energy to polymerize and form DNA. The lagging strand, or the DNA strand elongating away from the replication fork, is not as simple. A primer is put by the RNA and DNA polymerase III replicates the segment. It is synthesized in parts as a series of segments called Okazaki fragments. As the fragments are being made, DNA polymerase I replaces the RNA primers with DNA. Lastly, DNA ligase forms a bond between all of the fragments. The reason why the lagging strand is so discontinuous is because DNA is anti-parallel and replicates only from 5' to 3'. Since the lagging strand is 3' to 5', or in the opposite direction, it must be broken up into segments in order to fully replicate it.